Hello everyone, I am Dr. Naveshriyar, Assistant Professor in the Department of Crop Physiology, SR University. In this lecture, I will discuss about photoperiodism and its role in improving crop productivity. So, the content of this lecture is photoperiodism and a little discussion about discovery, and later we will discuss about short day, long day and day neutral plant. At the end, about floral hormone, how this hormone influence this photoperiodism. So first, let's understand about photoperiodism. First, try to understand this meaning. Photo period. Period means certain duration. Photo means light. So it is related to light duration. So the plant, in order to flower, it requires certain day length. So that day length is called as a photo period. So in responses to this photo period, changes in the flowering pattern of the plant is known as photoperiodism. Let's make it easy for you. So photoperiod means it's a relative length of day and night. It's just a duration. So photoperiodism means it is the response of plant to this relative length of day and night. So response of plant to this period or photo period is known as photoperiodism. So major scientists who plays a very important role in photoperiodism is Garner and Allard. So they worked on short day plant. So that too soya bean and tobacco, biloxi variety of soya bean and Maryland mammoth variety of tobacco. So they identified that just varying the night time or duration of night and day period, we can alter the flowering in short day plants. So basically, they worked first on tobacco plant. Among soya bean and tobacco, they worked first on tobacco plant. What they did means they kept tobacco plant under shade net. So those tobacco plants showed flowering. And what are the tobacco plants they kept under the light condition? It won't show flowering. So what it means so flowering requires long night time and short daytime. So tobacco plant is a basically a short day plant. So short day in the name itself is there. It requires a short day period and long night period. That too long uninterrupted night period. So what is this uninterrupted? What happens if night period is interrupted with any flash of light means in next slide we will know. So in this picture you will understand about the mechanism or properties of the short day plant and long day plant. So just observe, this is the 24 hours duration of day and night time. So for example, this line, middle line represents the 12 hours period or critical duration. So if night period is very less, day period is more. So that time short day plant won't show flower because name itself indicates short day plants requires a short day period. So in this second, so of course according to the definition, short day is there, long night is there. So that time the short day plant show flowering. So what is this then? So again daytime is very less, nighttime is long. Even though nighttime is long, they gave just a flash of light in the mid of dark period. So that time also short day plant won't show flowering. What it means then, even though we have short daytime, long night time, the short day plants are not flowering because short day plant requires uninterrupted dark period. So just interrupting this dark period with a flash of light affects the flowering in short day plant. So coming to long day plants, so of course name itself indicates that long day. Long day means more day and less night period. That time long day plants will show flowering. If the daytime is less, night time is more, then long day plant it won't show flowering. So here under short day condition, if you interrupt this night period, long night period with just a flash of light or like night break, So that break causes flowering in long day plant, but that break causes affect the flowering in short day plant. I hope you clear about short day and long day. Just according to name, short day plants requires short day, long day plants requires long day. Just vice versa. So these are the plants require relative short day plant and continuous or uninterrupted dark period. So that is known as short day plant. These are the examples. So just remember tobacco, soya bean, xanthium or cockleber. So these are the examples of the short day plant. So it requires 16 hours among 24 hours, 16 hours of dark, 8 hours of light. Short day plants will flower. If interruption of this dark period with flash of light inhibits flowering in short day plant. If the interruption of light period with dark or shading, so it won't affect the flowering in short day plant. Basic criteria is you can't disturb this dark period, you can alter this light period, no problem for short day plant, but this night period is plays a very important role in flowering. So coming to long day plant, name itself indicates that these plants requires longer daylight and shorter night time. So these are the some examples. What are the examples? Henbane, spinach, sugar beet, wheat, and barley. So all these are acts as a long day plant. So what do you mean by day neutral plant? It's not like it requires a equal day and night. It's like it is not affected much. It is almost insensitive to day and night period. It can flower in both long day and short day conditions. Some example for day neutral plant is tomato, cotton, sunflower, cucumber. So these are the examples of day neutral plant. Its range is different. So for example, here I mentioned one range 5 hours to 24 hours. If you give full light or full dark, 5 hours of light and remaining hours of dark, 19 hours of dark also, it will show flowering. These are the insensitive plants. So another classification is the major is short day, long day and day neutral. This is another classification, long short day plant and short long day plant. So long short day plant means these are the basically a short day plant, but initial days during its vegetative stage, it requires some days as a long day. So these are the plants which short long day means these are the basically a long day plants. It requires initially short day for its flowering. These are the short day plant, but it's exposed to long day during early period of growth in juvenile stage or in vegetative stage. When requires this long day exposure. So coming to short long day plant, these are the basically a long day plant must be exposed to short day during early period of its growth. So example is long short day plant, bryophyllum. Short long day plant is wheat and rye. Wheat acts as a long day also, sometimes it acts as a short long day. So then, of course, this one long day, short day, day neutral we discussed. How plants know is it long day or short day? So that is the perception of photoperiodic stimulus. Who receives the signal of this day period or night period means. So in the plant, leaf is the organ which perceives this photoperiodic changes. That relative length of day and night, whatever the changes is there, that is perceived by the organ of the plant called leaves. In so many exams they will directly ask what is the, or in the fill in the blanks dash is the organ responsible for the photoperiodic stimulus perception means leaf is the organ which perceives the photoperiodic stimulus. So as a result, the floral hormone also produced in the leaves, and after that it transferred to the inflorescence region or tip of the shoot or buds. So this one we need a experimental proof. We can't say that leaf is the organ which perceives generally. So they experimentally proved, they conducted so many different type of experiments to know that is it only leaf or any other organs also responsible. One by one we will understand. After this experiment they concluded that leaf is the organ which is responsible for this photoperiodic stimulus. So how they conducted this experiment means, so they grow on different pots with different characteristics. For example, in this picture, this is the normal plant. Cochleber means this is a xanthium plant and it is a short day plant. So in the figure A, what they did means just it is a full plant. They didn't do any changes. So they kept under short day condition. So it showed flowering. So what is B then? So in B, what they did, they removed all the leaves because they want to prove leaf is the organ which perceives the short day. That's why what they did means they removed all the leaf from this plant and exposed to short day. That time the plant didn't show any flower. So next stage in the C pot, what they did means they removed all the leaves except single leaf. This single leaf they exposed to short day condition. Single leaf also enough to transfer the stimulus. So this plant shows flowering. So then they concluded that of course leaf might be the organ which perceives the light. Same they did in long day condition because being it's a short day plant, under long day it won't show flowering. So here they removed all the leaves. Of course without leaves they won't show flowering. So here in the F pot what they did means all the leaves they exposed to long day and only one leaves they exposed to short day. So only by exposing one leaf to short day, it stimulates the flowering. So by this experiment, they concluded that leaf is the organ. Not only like this, from leaf, it is how we transfer this to one plant or how we transfer to inflorescence. Later, they conducted one more experiment. This is same, I think. Xanthium whether defoliate, same. They did, I think it's repeated. So next one, how they did means, so they took two branches. So they conducted in three pots with two branches. So one branch they exposed to two branched xanthium, one branch they exposed to short day, and another branch exposed to long day. So in both branches, in that time also, both branches they got flowering. It means if one branch exposed to short day also enough, that stimulus is transferred to another branch and stimulate the flowering. So that's why they concluded that there is a hormone which is synthesized in the leaf. That hormone is the responsible for flowering. That hormone, whatever it is synthesized in the leaves, it transfer from one place to another place and stimulate flowering in entire plant. This conclusion they got from this branching experiment. So in the next pot, what they did means they removed all the leaves. They kept only one leaves. For one leaf, they give short day treatment, and another one branch they exposed to long day conditions. Still, it showed flowering. In third one, they removed all the leaves and only for this without leafing branch they gave short day treatment. Another one is long day. In both the branches, they didn't got flowering. So it indicates that of course in previous experiment they already got one conclusion that leaf is the organ which perceives the stimulus, and in this experiment they got one conclusion that so leaf is the organ, but in that organ some hormone is synthesized which transfer from one part to another part. Because of that stimulus only the entire plant will show flowering. So again they did like this. So what they got, I think this one also repeated. So they got this experiment like, so FT or florizon. So they concluded that one hormone is there, flowering responsible hormone. They kept a name like we have so many hormones like auxin, gibberellin, cytokinin. These are the plant hormones, growth promoters. So like that, one hormone is there, that is florigen. Because of relative length of day and night, this hormone is synthesized in the leaves, and after that this hormones transferred to this meristematic region or inflorescence region, where it will convert it into the flowering stage. So then it initiate flowering in the tip of the shoot. So then they gave some details about the nature of the floral hormone. So they gave name like florigen. Then they conducted this grafting experiment. What is meant by grafting experiment means, here you see this is the short day plant. What they did, so first they exposed this one plant to short day treatment and it showed flowering. After that they cut down this one and then they joined with long day plants or short day under long day treatment. So then this plant also showed flowering. And another one is, if they exposed long day treatment, two long day treatment grafted together, it won't show flowering. But one long day treated short day plant and another one is short day treated short day plant, if they grafted, so both branches, like branching experiment, both edges got flowering. So then they concluded that floral hormone can be translocated from one plant to another plant, not only from one organ to another organ, it can transfer from one plant to another plant. For example, if one branch of the cochlear burr has been short day condition is grafted to another plant which kept under the long day condition, both of which have been kept under long day will not flower. So like this, if both kept under long day, they grafted together means it won't flower. But one long day, one short day grafted together means it will show flowering. So florigen is the plant hormone which synthesized in the leaf and translocated to another branch or another part. So then this is I think this completes about how they concluded leaf is the organ and how they got to know that florigen having hormonal nature. So coming to phytochrome, phytochrome is like a blue proteinaceous pigment. It is a pigment. So leaf, we have a lot of pigments, chlorophyll, carotenoids, so many pigments have in leaves. So like that, phytochrome is kind of pigment which perceives the light, certain light period, not all, because light contains so many radiations, violet radiation, red radiation, blue radiation, but phytochrome only captures the specific radiations and influence this photoperiodism. Phytochrome means it is a blue proteinaceous pigment and also it exists in two forms. What are those? Means okay, here I will tell you. Oh, so what are those means, there are two forms are there, one is PR form and another one is okay, so one is PR form and another one is PFR form. So what do you mean by PR and PFR means? This is the phytochrome red light, phytochrome infrared light means phytochrome which absorbs the red light absorbing form, and another one is infrared light absorbing form. So during daytime, so when there is a red light is more, during daytime red light will be more and during night time far red light will be usually far red light more. So during daytime PR form, red light form is converted into PFR form and this is the active form. It will either promote flowering or otherwise inhibit flowering. So this PFR is the active form. It directly sends signal to nucleus to initiate the florigen hormone genes, or it need to stop this florigen hormone genes. So the genes which are responsible for floragen synthesis is FT genes. So that gene synthesized by the signaling response of PFR. So the PFR produced during the daytime. So when PR form is converted into PFR. Just for your knowledge, you just understand. Phytochrome is the blue proteinaceous pigment. It exists in two forms: PR, red light absorbing form. PFR is the far red light absorbing form. During daytime, when there is a nanometer is there 660, and so in during 660 nanometer or during daytime it converts PR to PFR, and during far red light or 730 nanometer range, so it converted to PFR to PR. So this one is the PFR is the active form. It will give signal to nucleus to activate this FT gene, that is floragen synthesizing gene. So this is kind of mechanism phytochrome holds. So the phytochromes are the leaf pigment which is used by the plant to detect the period of light and dark. Organ is leaf, but these phytochromes are the leaf pigments like chlorophyll during photosynthesis, how it will absorb the light. Like that, phytochrome during daytime or nighttime, it absorbs these radiations and it influences the flowering. So the response of plant to relative length of day and night, of course it's photoperiodism. This response helpful by the phytochrome. So next, phytochrome discovery, Brothwick and Hendrick. So Garner and Allard discovered relative length of day photoperiodism classification, and Brothwick and Hendrick, they are identify this pigment responsible for photoperiodism is phytochrome. So here some explanation related to phytochrome. We already discussed this in the last slide. So the inhibition of flowering, this one I will tell you later. So just to remember, phytochrome exists in a two form. One is red light absorbing form, we call it as a PR, and far red light absorbing form, we call it as a PFR. Two forms are there. So that is phytochrome. So here its size is 250 kilodalton. It exists in homodimer form. So two identical polypeptide is there. So each weight about size of this each polypeptide is 125 kilodalton. So because of the sunlight contains more red light than the moonlight. So red light means daytime, far red light means nighttime. So here PR is the inactive form, active form is PFR. So during red light, whenever the nanometer range is 660, so that time PR form is converted into PFR. So far red light means PFR is converted into PR form. So like this, the conversion responsible for the flowering in long day and short day plants. So here I will explain one more theory. In short day plant flower when days are short. We already discussed. Short day means day is short, night is long. In short day plant PFR. So here what in the last slide. Wait. In the last slide we discussed that PR is there. So during daytime it converts to PFR. So during night time it converts to PR. So here in short day, when the daytime is very less, so the PFR, if it is more, PFR more means it clearly indicates that lighted period is more. So PFR is more means it clearly indicates that light period is more, because during light period only PR converted into PFR. This PFR concentration is increased means it will decrease the or it affects the flowering in short day plant. So it reverses in long day. If PFR concentration is more means it clearly indicates that day period is more. It will influence flowering in long day plant. So this reverse. So in short day plant PFR inhibits the flowering. Hence, the flowering requires the low level of PFR. In long day plants, the flower when days are long. So long means day is more, then concentration of PFR also more. Hence, it requires the night period used to be less than the critical length. So, in most of the exam, they will ask this question: PFR to PR ratio. PFR to PR ratio is high for long day plants flowering. So this PFR to PR ratio is less for flowering in short day plant. So because PFR inhibits flowering in short day plant and promotes flowering in long day plant, so there is no activity of PR. So highly active is PFR. Okay, this is the phytochrome. It converts in the red light, and for red light it converted back. So this one clearly we can understand. So this already we studied what it means. So during short day or long day, if light period is less, dark period is that too uninterrupted dark period means short day plants will flower, long day plants will become vegetative. So next condition is it is the long day, short night. That time short day plant vegetative, long day plant shows flowering. So here, even though it is a short day, means day period is less, night period is more, but night period is influenced by flash of light. That time short day plant won't flower, long day plant show flowering. This one is day period, again night period, daytime or light period, again dark period. So this alteration, if it is happens also, there is a more light period. That's why that short day plant is vegetative and long day plant show flowering. If there is a very less light, very less dark, means again very less light, very less dark. So dark period is reduced, almost equal light and dark also. Long day plants will flower and short day plants won't show flowering. So this one we already discussed. So then what is this means? Here you see, these are the long day plant, these are the short day plants. So whenever there is a less light time, night time, short day plant won't flower, long day plant show flowering. Whenever night period is more, short day plant flower, long day plant not flower. So here if it is interrupted by red light means it's like a daytime or light time. Using any light you can create or interrupt this dark period. That time short day plant won't flower, long day plant will flower. Here you see, so they interrupted this night period, but first they gave red light. Suddenly, so the red light means the PR is converted into PFR. That PFR inhibits the flowering in short day plant. But here what happens? So red light followed by they gave forward light. Forward light again converts so PFR to PR. So it inhibits the activity of PFR inhibition. That's why the end light is PFR means the short day plants flower, long day plant won't flower. Here red light followed by forward light again followed by red light. So red light means PR is converted into PFR. PFR concentration is very high. It inhibits flowering in short day and it promotes flowering in long day. Here again red forward, but the end the plant exposed to forward. Forward means dark period. So that time only individual radiation you are varying means if you end up at the end you expose to forward light means it will inhibit flowering in long day and it promotes flowering in short day. So I hope you understood. So because this one please remember, so PR don't forget. So PR is there, PFR. So during red light, so here is there now, during red light. So red light what happens? PR converts to PFR. It inhibits flowering in short day and it promotes flowering in long day, inhibits flowering in short day and promotes flowering in long day. So it reverses forehead light reverse this reaction. So if you expose to forehead light, the PFR converted back to PR. So here, if exposed to forehead light, PFR is converted to PR. So if PFR concentration is reduced, then short day plant will show flowering, and long day plant, it inhibits the flowering. That like this. This formula or this equation, don't forget. So then one more hormone is there, gibberellin. This gibberellin hormone, what they will do means, we have so many hormones, plant regulated hormones are there. Among this, the flowering hormone gibberellin is responsible for flowering in long day plants under short day condition. Even though short day is there, if you spray this gibberellin hormone, it will increase, because gibberellin is mainly responsible for cell elongation. So and also it will influence the synthesis of flowering hormone like florigen. So because of that, if you spray the gibberellin, so what happens means, in long day plants it show flowering even though it's a short day condition. So what do you mean by speed breeding then? So they are using same concept. They are exposing long day plants to almost 22 hours of light period and remaining 2 hours of dark period. Long day plants requires a long day and short night. So they are using LED lights. They are in a laboratory, they are giving this artificial 22 hours of light and 2 hours of dark. Because of that, the plant light requirement it fulfilled means, suddenly the plants complete its juvenile stage or vegetative stage, suddenly transform to reproductive stage. So number of breeding cycle you can improve just by using or altering this day and night. So it is highly useful in reducing the duration of the breeding cycle, because you want to release some varieties, you want to go for 6 to 7 generation. So to avoid that, for example, it's a 4 months 3 months old crop like wheat and barley, so you need to wait up to 4 months. Using this speed breeding, you altering this day and night time, you can get it in a very quick, and you reduce this breeding cycle, and you will get more number of yields or more number of population in a short time. So this one, they are using this photoperiodism response of long plants to day and night, they are using to create under speed breeding. So I hope you understood. First we discussed about what do you mean by photoperiodism. Later we discussed on short day plants, long day plants, day neutral plants, and after that what is the organ responsible means, leaf is the organ which is responsible, and which hormone synthesized in the leaf for flowering means florigen, and later in what purpose photoperiodism is helpful means, it is helpful for speed breeding. So coming to some MCQs on photoperiodism and phytochrome, in most of the exams under physiology part, they ask 1 or 2 questions from this photoperiodism. So let's solve few questions. So photoperiodism was first discovered by. First, in second slide, we discussed that photoperiodism categorized into short day, long day, and day neutral, and they worked on Garner and Allard. They worked on short day plant. That short day plants are Maryland mammoth variety of tobacco and biloxi variety of soya bean. And next question, short day plant flowers when? So night length exceeds the critical duration. Night length is short, day length is long. No, short day plan means short day means long night. So night length exceeds the critical duration is the answer. So next one is site of perception of photoperiodism. Organ of perception of photoperiodism. So what is the organ? Root, stem, leaf, flower means. So it's a leaf is the organ which is the perceives this relative length of day and night. Next one is flowering hormone is called. So which hormone we call it as especially flowering hormone means, so florigen, it responsible for the flowering hormone. This auxin, gibberellin, cytokinin also influence the flowering, but flowering hormone they gave florigen means you need to choose this florigen as an answer. Next one is active form of phytochrome. In PR and PFR, PFR is the active form. PFR inhibits flowering in short day plant. It promotes the flowering in long day plants. Another one is night break with red light. So promotes flowering in short day plant, inhibits flowering in short day plant, no effect, and converts short day plant to long day plant. This one not possible. And no effect, of course it will affect. Promote flowering in short day plant means no, it's wrong, because night break, it requires uninterrupted dark period. So if dark period is breaking because of red light, then it inhibits flowering in short day plant. If you give red light followed by forehead light means okay, it will promote flowering in short day plant means no effect, that much no effect. So here they mentioned only red light, so red light will inhibit flowering in short day plant. So in short day plant, a long night is interrupted by, we discussed in this question, no? Same thing here is there. In short day plant, long night is interrupted by red light, okay, followed by immediately by forehead light. Whatever the conversion is there, so whatever the conversion is there, PR to PFR, whatever the conversion is there under the red light, so red light, it is nullified by forehead light. So it's reversed by forehead light. So because of that, it causes no effect. So the flowering is inhibited, flowering occurs normally, flowering is delayed, no, not delayed or inhibited. So the flowering occurs, no effect. Normally it will occur. So when red light is followed by immediately forehead light, so then only this effect will nullify. So next one is first plant used in photoperiodism. We know that scientists who worked on photoperiodism is Garner and Allard. They worked on both tobacco and soya bean. But first plant among this means it's a tobacco. If they gave soya bean, please don't confuse. So first plant among soya bean and tobacco, first plant they worked on photoperiodism means it's a tobacco plant. I think hope you got some knowledge regarding photoperiodism, because it's highly important topic. Just don't forget this definition. Photoperiodism entirely related to the flowering of the plant. The definition of photoperiodism means it's a relative length of day and night, so which is responsible for flowering of the plant. So the classification is short day, long day, day neutral. I hope you got it some information. So okay, thank you so much.